In 2023, a neuroscientist ran the numbers on T-Rex. Not the bones, not the teeth, not the footprints. The neurons. And what she found caused a war inside paleontology that is still being fought today. Because if her calculation was right, T-Rex didn't just have a bigger brain than we thought. It had neuron counts comparable to a baboon. Not a lizard, not a crocodile. A primate. The field erupted. Some researchers called it a revolution in how we understand the most famous predator that ever lived. Others called it one of the most dangerous overclaims in the history of paleontology. And right in the middle of that fight, new imaging technology did something that changed the terms of the entire debate. It looked inside a 500-million-year-old fossil and found a preserved nervous system, intact in stone 500 million years old. What that discovery revealed about the origin of animal intelligence and what it means for how we read dinosaur brains is something no textbook has caught up with yet. In the next 15 minutes, that changes. To understand why the T-Rex neuron debate matters and why it exploded the way it did, you need to understand the fundamental problem with studying dinosaur intelligence. Brains don't fossilize. Soft tissue almost never survives. The organs that tell you how an animal thought, the structures that encode memory, process sensory information, generate behavior, they decompose. They vanish, leaving behind only the hard shell that contained them. For most of paleontology's history, that meant one tool and one tool only. The endocast. An endocast is exactly what it sounds like, a cast of the interior of the skull cavity, physical or digital. It captures the approximate shape of the brain and allows researchers to measure volumes, identify regions, and compare structures across species. It's a powerful tool, but it has a serious limitation. The endocast is not the brain, it's the room the brain lived in. And in reptiles, in crocodilians, in many theropods, the brain doesn't fill the skull cavity the way a mammal brain does. There's fluid, there's tissue, there's space. Which means endocast volume can overestimate brain size, sometimes significantly. This single methodological problem sits at the center of almost every disagreement in dinosaur intelligence research. But in the last 5 years, the toolkit expanded dramatically. High-resolution CT scanning now allows researchers to map not just the shape of the skull interior, but the density, the texture, the fine structural details of bone, and any preserved material within it. Synchrotron imaging uses extraordinarily intense X-rays to map internal chemistry at extremely high resolution. And in 2025, that technology produced something extraordinary. In 2 minutes, you'll see what it found. But first, the controversy that made everyone realize the stakes. Neuroscientist Susanna Herculano-Husel, one of the world's leading experts on brain scaling and neuron counting, turned her methodology on theropod dinosaurs. Her approach was not based on endocasts. It was based on a mathematical model relating brain mass to neuron count across hundreds of living vertebrate species. She applied that model to estimated theropod brain masses. And the number that came back for T-Rex stopped the field cold. Her calculations suggested T-Rex may have possessed telencephalic neuron counts, neurons in the higher cognitive processing region of the brain, in the range of some living primates. Baboon level, possibly higher. Not the dim, reactive predator of popular imagination, but an animal with the neural substrate for learning, long-term memory, behavioral flexibility, and potentially social intelligence. She argued provocatively that T-Rex may have used tools if it had the right forelimbs, that it may have had a cultural life, that its behavioral repertoire could have been, in her words, closer to a Komodo dragon crossed with an elephant than to a giant crocodile. The response from the paleontological community was immediate. And it was not gentle. In 2024, Kai Kasper and colleagues published a direct rebuttal. Their core argument, the neuron scaling model Herculano-Husel used, was calibrated on living birds and reptiles, animals whose brain-to-neuron ratios we can measure directly. Applying it to extinct dinosaurs required assuming that theropod brains scaled the same way. And that assumption, Kasper's team argued, was almost certainly wrong. Because birds, the living dinosaurs, have unusually neuron-dense brains precisely because of evolutionary pressures specific to flight, to miniaturization, to the demands of aerial life. Pressures that T-Rex, a massive ground-based predator, never experienced. The model was being applied outside its valid range. The actual neuron counts, Kasper's team estimated, were likely significantly lower. Not primate level, not baboon territory. But also, critically, not the dim reptile either. Here's what both sides agree on. T-Rex had a larger, more complex brain than its reputation suggests. The olfactory bulbs, the regions processing smell, were proportionally enormous, likely surpassing the olfactory capability of most living mammals. The optic lobes were substantial. The auditory processing regions were well developed. The disagreement isn't about whether T-Rex was sophisticated. It's about how sophisticated, and what framework we're allowed to use to measure an animal that has been extinct for 66 million years. That debate is still open. And the imaging technology that may finally resolve it just produced its most important finding yet. In 2025, Jakob Winther and colleagues published a study in Science Advances that demonstrated something paleontologists had long considered essentially impossible. They found a fossilized nervous system. Not the skeleton, not surface impressions. A preserved ventral ganglion, neural tissue, identifiable in structure and chemistry. Using synchrotron imaging, they mapped not just the shape of the neural tissue, but its chemical composition. The chemistry matched nervous system material. The structural organization matched a ganglion-like neural cluster. Individual nerve bundles were identifiable. An animal that lived 500 million years ago had left behind a readable map of its own nervous system. Now this is a Cambrian animal, not a dinosaur. The specimen isn't T-Rex. But here's why it changes the dinosaur brain debate completely. The Cambrian is the origin point. It's where complex animal body plans first appear. Every vertebrate alive today, every fish, every reptile, every dinosaur, every mammal, every human traces its neural architecture back to this period. What Winther's team proved is that the tools now exist to read neural tissue in ancient fossils. Tissue that was previously assumed to be irretrievably lost. Apply that technology forward in time. If we can detect and chemically identify a preserved ganglion in a 500 million year old Cambrian specimen, what becomes possible when we apply the same methodology to fossils from 100 million years ago? To theropod skulls with better preservation than anything previously examined. To the specimens where the endocast debate has been loudest, and where a direct chemical map of preserved neural material could settle the question of brain volume, brain density, and neuron distribution once and for all. The T-Rex neuron debate has been fought with indirect evidence. Synchrotron methodology just handed both sides a potential path to direct evidence. The next decade of dinosaur brain research will be built on this foundation. While the neuron-count debate was erupting, a separate team was asking a different question entirely. Not how many neurons did adult dinosaurs have, but how did their brains develop? Research published in Nature Communications in 2024 by King and colleagues examined the endocranial development trajectory in non-avian dinosaurs, mapping how the brain cavity changed shape across growth stages in multiple species. What they found reframes the intelligence question at a fundamental level. In many theropod species, the brain didn't just grow larger as the animal matured. It changed shape. Specific regions expanded disproportionately relative to others, and the regions that showed the most dramatic proportional growth were not the regions associated with basic motor function. They were the regions associated with sensory integration and higher processing. In living animals, disproportionate growth in sensory and cognitive processing regions during development is often associated with behavioral complexity. It's the neural signature of an animal whose survival depends on learning from experience, not just running hardwired instinct programs. You see it in corvids, in elephants, in great apes. And now in theropod dinosaurs. This is where the endocast versus neuron-count debate starts to look like the wrong argument. Because even if the absolute neuron count is lower than Herculano Husserl proposed, even if Casper's rebuttal is substantially correct, the developmental trajectory evidence suggests theropod brains were organized for behavioral flexibility in ways we haven't fully accounted for. The issue isn't just how many neurons. It's how those neurons were arranged, which regions were prioritized, what the brain was built to do. And that question, the architectural question, is one the endocast record, properly read, can actually begin to answer. Before we go further, there's a warning label that every brain inference in paleontology needs attached to it. And it comes in the form of the strangest animal ever found in Illinois. Tullimonstrum gregarium, the Tully monster, state fossil of Illinois, found in 1958, 307 million years old. It has eyes on rigid stalks, a proboscis tipped with a tiny claw, a torpedo-shaped body unlike almost anything in the fossil record. For decades, no one could classify it. Then in 2016, a major study declared it vertebrate, specifically related to jawless fish, one of our distant relatives. In 2017, Lauren Salin and colleagues published a systematic dismantling of that conclusion in Paleontology. The structures identified as vertebrate features, the notochord, the gill pouches, the neural structures, were reanalyzed. Salin's team found that the identifications relied on preservation artifacts and interpretive assumptions that didn't hold up under scrutiny. The Tully monster, they argued, was not a vertebrate. Its position on the tree of life remains unresolved today. Here's why this matters for dinosaur brains specifically. If we cannot confidently classify an exceptionally well-preserved fossil from only 307 million years ago, using all the tools available to modern paleontology, what does that tell us about the confidence we should place in brain inferences made from skull cavities? It tells us endocast shape is not brain shape, estimated volume is not neuron count. Neuron count models calibrated on living animals may not apply to extinct ones. Every claim in this field, including the exciting ones, carries an asterisk. The asterisk isn't a reason to dismiss the science. It's a reason to pay attention to which evidence is direct and which is inferred. Now, with that humility calibrated, let's go back to the dinosaur skulls where the evidence is actually strongest. Let's stack what we actually know directly from the evidence. Not inference chains, not model projections. What the fossils themselves, read with modern tools, actually show. First, olfaction. Across multiple theropod species, Tyrannosaurus rex, Velociraptor, Troodontids, the olfactory bulbs are disproportionately large. Not large for a reptile. Exceptionally large among theropods and very large by vertebrate standards. We're talking about animals that could detect and track scent information at a level that rivals many top modern trackers. The olfactory processing power embedded in theropod neuroanatomy was exceptional by any comparative standard. This is not a modeling assumption. This is a structural measurement from endocasts. It is as close to direct evidence as this field gets. Second, vision. The optic lobes in large theropods were substantial, and in the smaller, faster theropods like Troodontids proportionally enormous. Binocular vision, the overlapping forward-facing fields that enable depth perception and precise targeting, was present in multiple theropod lineages. You don't evolve high-resolution binocular vision unless your survival depends on precise visual judgments. That means active hunting strategies requiring real-time spatial computation, not reactive ambush, not passive opportunism. Third, the telencephalon. This is the contested territory, where Herculano Husel and Casper disagree. But even Casper's more conservative estimates place theropod telencephalic development above the crocodilian baseline. The ceiling of the debate is primate-level complexity. The floor is still a brain meaningfully more sophisticated than we gave these animals credit for. And the King et al. Developmental trajectory work adds a dimension neither side of the neuron debate fully accounts for. The architecture was being organized, from early development, for sensory integration and behavioral flexibility. Stack those three findings together. Exceptional olfaction, high-resolution binocular vision, a telencephalon larger and better organized than the reptilian baseline, a developmental trajectory pointing toward behavioral flexibility, and a new imaging methodology demonstrated by Vinther's Cambrian nervous system work that may soon allow direct chemical detection of preserved neural tissue in dinosaur skulls. T-Rex wasn't a giant crocodile on legs. It was an apex sensory predator, navigating its world through overlapping streams of rich sensory information, capable of learning, capable of memory, operating a brain that 500 million years of evolution had been refining since the Cambrian. We just didn't have the tools to see it clearly. Until now. Here is the thought I want to leave you with. The entire debate we've covered today, the neuron count war, the endocast limitations, the Tully monster warning, the Cambrian nervous system, it's all downstream of one fundamental truth. We are trying to read the minds of animals that no longer exist, using the shells they left behind. Every endocast is a room with the furniture removed. Every neuron count model is a map drawn from a different continent. Every fossil is a message written in a language we're still learning to read. And yet, imperfect as these tools are, they keep pointing the same direction. The dinosaurs that ruled this planet for 165 million years were not the slow, dim, autopilot creatures that ended up in a century of bad movies. They were sensory predators, behavioral learners, animals running ancient, refined neural hardware that traced its origins back to the Cambrian, to creatures so strange we still argue about what they were. The more precisely we look, the more sophisticated the picture becomes. And the most precise tools we have ever pointed at a dinosaur skull are being turned on the specimens right now. Whatever they find, it will not make these animals less impressive. Subscribe because when those results come in, you'll want to already know the story behind them.